Picture a single cell under the microscope, tiny, ordered, alive. Now zoom out to forests, oceans, and the human body itself. Biology stretches from the invisible to the immense, but what rules tie all these scales together? Welcome to Rest Academy. Tonight you'll travel through the complete map of biology explained in a calm, continuous flow from molecules and genetics to evolution and ecosystems, asking what life is, how it sustains itself, and where its boundaries might end. To make this journey complete, I've prepared a free practical guide with a concise summary of tonight's lesson, waiting in the description like a map you can return to whenever needed. Settle in, let your mind rest, and let's begin. You encounter the problem immediately. To define life, you know it when you see it, yet every definition collapses at its edges. A stone is stable but inert. A flame grows and consumes but leaves no descendants. A virus replicates but only by hijacking cells. So where does biology begin? The cell is the standard answer. Defined as the smallest unit of life, a cell is a compartment enclosed by a membrane capable of maintaining itself and reproducing. Within, intricate molecules interact with precision. They use energy, repair damage, and store coded information. This ability to sustain order in the midst of entropy makes cells more than chemistry. Still, biology resists such neat closure. Consider prions, those misfolded proteins that propagate by inducing other proteins to twist into their shape. They lack DNA, RNA, or membranes, yet they spread, and in doing so, they blur the borders of what counts as alive. Viruses do something similar on a larger scale. They evolve, adapt, and even develop sophisticated strategies, but without a host, they are inert particles of nucleic acid and protein. To understand life, you begin not with a rule, but with a spectrum. At one end lies raw chemistry, at the other complex organisms. Cells occupy the middle, bridging matter and vitality. On Earth, they appeared about 3.5 billion years ago, long after the planet cooled but long before multicellular creatures. For most of Earth's history, life meant microbes, bacteria, and archaea, tiny yet versatile engines of survival. One image helps here. Imagine a map. At its center is the cell, the hub. Around it stretch paths leading to molecular biology, genetics, ecology, evolution. Each path illuminates one aspect of what cells can do. The challenge is that no path can be walked alone. Every system in biology connects back to that central hub. A case illustrates the stakes. When doctors examine a patient with a suspected viral illness, they must decide whether the cause is an organism that can be treated with antibiotics or a virus that cannot. Misjudging this boundary between cellular life and parasitic particle carries consequences. A virus cannot be killed in the usual sense, and attempting to treat it as if it were a bacterium leads to failure. Thus, the definition of life is not philosophical alone, it carries clinical weight. Reflect for a moment, and asking what life is, you confront not only the mystery of beginnings but also the practical puzzles of daily existence. It is a question both ancient and immediate. If a cell is the unit of life, what hidden steps carried chemistry across the border into that first living cell? To approach the first cells, you must imagine an Earth without green forests, animals, or oxygen. The young planet, over 4 billion years ago, held seas infused with minerals, a sky veiled in volcanic gases, and energy streaming from sunlight, lightning, and geothermal vents. This backdrop supplied the ingredients for a profound transformation, chemistry becoming biology. One useful picture is a shoreline where waves repeatedly concentrate simple molecules in pools that evaporate under the sun. These molecules include amino acids, nucleotides, and lipids, basic building blocks that can form more elaborate structures when pressed together by cycles of wetting, drying, and heating. In such pools, membranes might self-assemble from fatty acids, enclosing drops of solution. A primitive container emerges, a forerunner of the cell. But a membrane alone is not life. Inside, there must be order, information, and reactions that sustain themselves. Early hypotheses proposed that RNA, a cousin of DNA, was central. Unlike DNA, RNA can both store information and act as a catalyst. Short strands of RNA may have copied themselves, albeit imperfectly. Errors in copying introduced variation, allowing a crude form of selection. In this way, pools of molecules gain traits of life, inheritance, mutation, and competition. Another model emphasizes metabolism before genetics. In this view, networks of chemical reactions arose near deep sea vents where hot fluids met cold seawater. Minerals in vent walls provided surfaces that guided reactions, producing energy-rich compounds. Over time, these networks may have become enclosed within membranes, later joined by information molecules. Whether genes came first or metabolism came first remains unresolved. Both lines of thought suggest that life is less a single spark and more a gradual assembly of parts that together became self-sustaining. The timeline was immense. From the cooling of Earth's crust to the appearance of robust cellular life, hundreds of millions of years passed. During that time, molecules learned to cooperate. Proteins formed from chains of amino acids, folding into shapes that accelerated reactions. Lipid membranes became more stable, selectively letting nutrients in and waste out. Information polymers refined their sequences to carry templates for proteins. The protocell, a hypothetical ancestor, was not yet as efficient as today's bacteria, but it marked a threshold crossed. Imagine a relay race. The baton passes from geology to chemistry, then from chemistry to biology. Rocks erode to release minerals. Minerals drive reactions. Reactions form molecules that join into systems. Systems cross into entities that replicate. Each stage depends on the one before it, yet adds a new layer of complexity. The first true cells were the runners who grasped the baton and kept moving. Evidence is scarce, yet microfossils in ancient rocks and chemical signatures in sediments suggest that by 3.5 billion years ago, bacteria-like organisms were already abundant. These pioneers transformed Earth. Some developed photosynthesis, capturing sunlight and releasing oxygen. Others mastered fermentation or chemosynthesis, extracting energy from minerals. Life diversified, not through sudden invention, but through steady adaptation to varied niches. A clinical example shows why these origins matter. In hospitals, researchers sometimes design lipid vesicles, artificial bubbles that mimic cell membranes to deliver drugs. Their stability, permeability, and self-assembly echo the very principles that gave rise to protocells. By studying how life may have begun, modern science learns how to create tools that aid medicine. Yet the ancient question persists: where exactly did chemistry end and biology begin? Pause briefly. You are tracing events older than mountains, yet their echoes persist in every cell of your body. Each heartbeat and breath continues processes that began in those primeval pools. If the first cells were born from chemistry, what hidden order allows their inner parts to work together without collapsing into chaos? Inside the earliest cells, you find a dense world of molecules that never rest. Among them, a thread-like polymer called DNA holds patterns written in a 4-letter code. DNA, or deoxyribonucleic acid, is a stable molecule designed to store information across generations. It does not act alone. Its instructions are transcribed into RNA, ribonucleic acid, which then guides the assembly of proteins, the versatile machines of the cell. This constant flow from DNA to RNA to protein defines molecular biology. Think of DNA as a library, RNA as a photocopy of a single page, and proteins as the workers who follow the instructions to build or repair. Proteins fold into intricate three-dimensional forms, their shapes suited to cutting, joining, or transporting molecules. Without them, reactions would proceed too slowly for life. Molecular biology studies how these three partners, DNA, RNA, and proteins, interact in networks of control and cooperation. The process begins with transcription. Enzymes unwind DNA and copy a gene into messenger RNA. This RNA strand then travels to ribosomes, structures built of RNA and protein themselves. There, translation occurs. Each triplet of RNA bases corresponds to an amino acid, which is added to a growing chain. The chain folds into a protein, guided by chemical forces. But this is not a simple one-way street. Feedback loops regulate expression. Proteins may block or enhance transcription of certain genes, ensuring that a cell responds to its environment. For instance, bacteria exposed to lactose produce enzymes to digest it only when lactose is present. This economy of effort reflects an ancient logic: conserve energy, respond quickly, survive. Errors and copying add another layer. Mutations, changes in DNA sequence, can alter proteins. Some mutations prove harmful, leading to disease. Others are neutral. A few confer advantage, fueling evolution. Thus, molecular biology explains not only daily cell function, but also the long arc of life's diversity. You can visualize this system as a factory with conveyor belts. DNA provides the master blueprint. RNA carries individual orders. Proteins act as specialized machines, cutting, folding, transporting. Supervisors, themselves proteins, adjust the speed of each line. The result is a dynamic balance, precise enough to maintain identity, yet flexible enough to adapt. A real-world illustration lies in cancer research. Cancer arises when control of gene expression fails. Mutations activate growth-promoting genes or silence repair mechanisms, leading to uncontrolled division. By tracing the molecular steps, mutations, misfolded proteins, disrupted feedback, researchers seek interventions. This is not merely theory, it directs diagnostics, predicts outcomes, and guides therapy development. Reflection is natural here. In every cell of your body, millions of reactions unfold each second without conscious command. You are alive because these networks run faithfully, silently, ceaselessly. Yet a question emerges: if proteins depend on precise shapes to act, what ensures that they fold correctly, and what happens when they do not? Proteins, nucleic acids, and other biomolecules are not only defined by their chemical composition, but also by their shapes. This is the domain of structural biology, the study of how form dictates function. A single twist or bend in a molecule can alter its entire role in the cell. Imagine a key fitting into a lock. If the ridges on the key change even slightly, the lock will not turn. In cells, enzymes act as locks and their substrates as keys. The enzyme structure creates a pocket where a specific molecule fits. When shape and chemistry align, the reaction proceeds rapidly. When they misalign, the process halts. Thus, structural biology explains why life depends on precise three-dimensional architectures. Proteins begin as chains of amino acids. Once built, they fold spontaneously, guided by interactions between side groups. Some attract water, others avoid it. Some form hydrogen bonds, others ionic bridges. The result is a compact form with loops, sheets, and spirals. But folding is not always perfect. Cells use chaperone proteins to guide the process, shielding fragile chains until they reach the correct configuration. When folding goes wrong, diseases may emerge. Misfolded prion proteins, for example, induce nearby proteins to adopt the same abnormal shape, leading to devastating neurological decline. Structural biology also examines nucleic acids. DNA is a double helix, with complementary strands winding around one another. This shape ensures stable storage of genetic information and precise replication. RNA, though similar in composition, folds into varied structures that allow it to catalyze reactions or regulate genes. The elegance of these forms underlines a central truth: structure is inseparable from function. To study these minute architectures, scientists use methods that borrow from physics. X-ray crystallography shines beams through purified crystals of proteins, revealing patterns that can be converted into atomic maps. Cryo-electron microscopy, by freezing samples at ultra-low temperatures, captures molecules in near-native states. Nuclear magnetic resonance provides yet another angle, allowing dynamic views of proteins in solution. Each technique unveils the invisible, showing how a molecule's bends and twists correspond to its actions. One metaphor clarifies this further. Think of a symphony orchestra. Each instrument has a particular shape that produces a distinct sound. A violin differs from a trumpet not only in material but in form. Similarly, biomolecules sound their functions through their folds and patterns. If one instrument is warped, the harmony suffers. Structural biology is the discipline that listens for these distortions and explains their consequences. A clinical context demonstrates its importance. Consider sickle cell disease. A single mutation changes one amino acid in hemoglobin, the protein that carries oxygen in red blood cells. This tiny substitution alters hemoglobin's shape, causing molecules to stick together. The cells distort into rigid crescents, clogging vessels and impairing oxygen delivery. The lesson is clear: microscopic structural changes can reshape entire lives. As you reflect, you realize that your body is a gallery of invisible sculptures. Their forms shift and sway, enabling thought, movement, and memory. But if structure is so vital, then how do these molecular sculptures interact chemically, and what underlying reactions sustain their dance? Life reduces to countless chemical reactions occurring in orderly fashion inside cells. This is the province of biochemistry, where molecules are not only described but also understood as participants in chemical cycles. Each reaction follows the same rules as in any laboratory flask, yet within cells they are organized into pathways that sustain life. At the foundation lie the molecules of life: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates, such as glucose, provide energy through breakdown into smaller units. Lipids form membranes and act as energy stores. Proteins, composed of amino acids, serve as catalysts and structural elements. Nucleic acids, DNA and RNA, store and transmit information. Biochemistry studies not just their identities, but how they react with one another. Central to this is metabolism, the sum of all chemical reactions in a living system. Metabolism divides into catabolism, the breaking down of molecules to release energy, and anabolism, the building of molecules that requires energy. Catabolism produces molecules like ATP, adenosine triphosphate, which acts as the cell's energy currency. Anabolism uses that currency to build proteins, nucleic acids, and membranes. The continual cycle between breaking down and building up is what maintains cellular life. Enzymes lie at the heart of this process. These proteins lower the energy barrier of reactions, allowing them to occur rapidly under mild conditions. Without enzymes, most reactions essential for life would be far too slow. Enzymes are specific, binding only particular substrates, and are regulated so that pathways remain balanced. For example, glycolysis, the pathway that breaks down glucose, involves 10 enzyme-catalyzed steps, each controlled by the needs of the cell. Pathways interconnect into networks. The citric acid cycle, for instance, receives products from carbohydrates, fats, and proteins, then feeds into pathways that generate ATP. This interconnection ensures that if one nutrient is scarce, others can substitute. Such flexibility allows organisms to survive in varied environments. A powerful image here is a city's economy. Raw materials enter factories, processed goods are distributed, waste is recycled, and energy fuels every step. Taxes and regulations control flow, preventing chaos. In cells, molecules flow through pathways, enzymes act as regulators, and energy supply sets the limits. Biochemistry, like economics, seeks to understand these flows and controls. Modern techniques reveal reactions in extraordinary detail. Mass spectrometry identifies metabolites by weight. Isotopic labeling tracks molecules as they move through pathways. Fluorescent probes illuminate reactions inside living cells. These methods show biochemistry not as static but as dynamic, with molecules constantly moving, joining, and breaking apart. Clinical cases highlight why this knowledge matters. In diabetes, for example, glucose cannot be properly taken into cells due to problems with insulin. The biochemical consequence is elevated blood sugar, which damages tissues over time. Understanding the molecular pathways clarifies why symptoms occur and guides strategies for monitoring and prevention, even if therapy lies outside the scope here. Pause to reflect. The ordinary act of breathing sustains biochemical chains that stretch back billions of years. Each molecule of glucose you consume enters pathways honed by evolution, ensuring your cells remain alive. Yet a deeper question remains: if life is chemistry in motion, what role do the fundamental laws of physics play in shaping and constraining these reactions? When you examine life at its smallest scales, physics quietly shapes every event. Biophysics applies the laws of mechanics, thermodynamics, and electromagnetism to biological systems, revealing why molecules interact the way they do. Even quantum phenomena, once thought irrelevant to biology, now appear to influence essential processes. Start with membranes. These thin barriers made of lipids are held together by hydrophobic interactions, a physical principle where water-repelling tails cluster away from the aqueous environment. Their flexibility, curvature, and ability to fuse or divide all follow mechanical rules. Proteins embedded in these membranes respond to forces, opening channels that let ions flow according to electrical gradients. This coupling of structure and force explains how nerves transmit signals at remarkable speed. Thermodynamics governs biochemical reactions. For a process to proceed, energy must decrease in the system or be coupled to another reaction that releases energy. ATP hydrolysis, the splitting of a phosphate bond, provides this push, making otherwise unfavorable reactions possible. In essence, life borrows from physics. It cannot violate thermodynamic laws, but it uses clever strategies to harness them. Consider protein folding once again. The reason a chain of amino acids collapses into a functional shape lies in energy minimization. The folded state has lower free energy than the unfolded one. Though the path may involve temporary misfolded intermediates, biophysicists measure these folding landscapes using spectroscopy and single-molecule force experiments, watching how proteins search for stability within physical constraints. Quantum biology offers even more surprising insights. In photosynthesis, for example, energy absorbed from light appears to travel through pigment molecules via quantum coherence, sampling multiple pathways simultaneously before settling on the most efficient. Similarly, in enzymes, quantum tunneling may allow subatomic particles to cross barriers they could not surmount, classically accelerating reactions beyond expected rates. These effects suggest that life has adapted to exploit quantum principles that normally fade at larger scales. A helpful image is that of a stage play. Chemistry writes the script, but physics builds the stage, controls the lighting, and dictates the rules of movement. Without the stage, the actors could not perform. Biophysics shows how the unseen architecture of forces enables the drama of life. One striking clinical connection is medical imaging. Techniques like magnetic resonance imaging, MRI, arise from physical properties of atomic nuclei and magnetic fields. By applying radio waves and measuring responses, physicians obtain detailed pictures of tissues without surgery. Here physics directly supports diagnosis, translating subtle atomic behaviors into macroscopic understanding of health. Reflect for a moment, even your thoughts rely on voltage changes across membranes, the same physical principles that govern sparks in a storm. Life does not stand apart from physics, it is physics shaped into resilience and complexity. But if physics provides the stage and chemistry directs the play, then how does the script of inheritance, DNA itself, ensure continuity and diversity across generations? Genes are sequences of DNA that encode instructions for building proteins or regulating processes. Genetics is the discipline that studies how these sequences are organized, expressed, and passed from one generation to the next. At its core lies the question of how information persists while still allowing change. Each cell in your body, aside from a few exceptions like red blood cells, contains essentially the same genome, yet different cells perform distinct roles. A neuron transmits signals, a muscle fiber contracts, a skin cell forms a barrier. This variety arises not from different genetic content, but from differences in gene expression, whether a gene is turned on or off, and how strongly epigenetic mechanisms such as chemical tags added to DNA, or proteins that package it, regulate these switches without altering the sequence itself. Inheritance follows patterns first described by Gregor Mendel. He observed that traits segregate and recombine according to predictable ratios, which we now know reflect the behavior of chromosomes during meiosis, the specialized cell division that produces gametes. Each gamete receives one copy of each chromosome, and fertilization restores the full set. This mixing produces both stability and variation. Mutations add further diversity. Some mutations arise spontaneously during DNA replication when polymerases insert the wrong base. Others come from external sources such as radiation or chemicals. Most are neutral or harmful, but a few offer advantages that natural selection preserves. Over evolutionary time, the accumulation of such mutations drives adaptation. Population genetics extends this view to groups. It examines how gene frequencies change under forces like selection, genetic drift, migration, and mutation. Even within one species, genetic variation can be immense, shaping responses to environment and disease. For example, variations in hemoglobin genes confer resistance to malaria, but also cause sickle cell disease when inherited in certain combinations. This illustrates the trade-offs encoded in our DNA. You may picture DNA as a library with billions of letters, yet evolution does not rewrite it in neat chapters. Instead, errors, recombinations, and insertions accumulate like edits in a manuscript copied endlessly by hand. Some edits distort meaning, others add nuance, and some produce entirely new chapters. Genetics seeks to decipher this manuscript, to read both the stable script and the variations that create diversity. In clinical practice, genetics informs both diagnosis and prognosis. A patient with a family history of a heritable condition may undergo testing to identify mutations. The presence or absence of specific changes can clarify risk and guide monitoring. For example, mutations in BRCA genes increase susceptibility to certain cancers, while treatment decisions extend beyond this discussion. Recognizing genetic predispositions demonstrates the real-world significance of inheritance. Pause to reflect. Within every cell of your body lies a history, not just of you, but of ancestors stretching back billions of years. Each gene carries echoes of survival, adaptation, and change. Yet the genome is vast, and to uncover its patterns requires tools beyond intuition. How can such immense data be interpreted without drowning in complexity? The genome of even a simple bacterium contains millions of bases, and that of humans holds more than 3 billion. To make sense of such immensity, biology turns to computational sciences. Bioinformatics applies algorithms and software to analyze genetic data, while biomathematics uses mathematical models to describe biological processes. Together, they reveal order hidden in overwhelming detail. Imagine a library so large that no person could read it in a lifetime. Computers act as readers, scanning entire genomes in seconds, aligning sequences and highlighting similarities. Through this, scientists can identify genes linked to traits, predict protein structures, and trace evolutionary relationships. For example, comparing viral genomes during outbreaks allows researchers to map how strains spread and mutate, guiding public health responses. Mathematics adds another dimension. Equations describe how genes interact, how populations evolve, or how molecules diffuse through tissues. Models can simulate epidemics, predict outcomes of genetic drift, or estimate how quickly resistance to antibiotics may spread. These models do not replace observation, but provide frameworks to test hypotheses and forecast scenarios. Consider gene expression data. Thousands of genes may turn on or off in response to a single stimulus. Bioinformatics organizes this into networks, showing which genes rise or fall together, suggesting pathways under shared control. Biomathematics then models how these networks behave, whether they stabilize in steady states or shift into new patterns. This integration transforms raw data into understanding. A metaphor clarifies. Think of a complex transportation system. Each vehicle represents a gene or protein, each route a pathway, and traffic patterns reflect interactions. Observing traffic at a single intersection tells little. Only by analyzing all routes together can one predict congestion or flow. Computers and mathematics provide the aerial view, showing how the system functions as a whole. Practical applications abound. In medicine, bioinformatics identifies mutations associated with disease, enabling targeted monitoring. In agriculture, it reveals genes that enhance crop resilience. In ecology, it tracks biodiversity by sequencing environmental DNA found in soil or water. Each case transforms raw letters of nucleic acids into actionable knowledge. One clinical example highlights its impact. When a patient's tumor is sequenced, bioinformatic analysis may reveal specific mutations that drive growth. Although treatment decisions fall outside this scope, the ability to categorize the tumor at the molecular level allows physicians to refine diagnoses and consider tailored approaches. This illustrates how computation bridges basic science and patient care. Reflection arises. Biological complexity is no longer beyond reach. With algorithms and models, patterns that once seemed inscrutable now emerge as clear signals. Still, the more data we gather, the more questions appear, as if each answer opens new corridors in the library. If genomes and models illuminate potential, then how does a single cell guided by such instructions grow and differentiate into a complete organism? You began as a single fertilized cell, no larger than the head of a pin, yet within that cell lay the entire potential for an adult body of trillions of cells. This transformation from one to many is the field of developmental biology, which examines how organisms grow, differentiate, and form structured bodies. It asks a profound question: how do identical copies of DNA give rise to such diverse tissues and organs? At the core is cell division. The fertilized egg undergoes mitosis, producing 2 cells, then 4, then 8, and so on. But soon differences emerge. Cells in distinct positions begin to receive varying signals, activating different sets of genes. Some cells specialize as muscle, others as neurons, others as skin. This process of differentiation transforms a uniform mass into a mosaic of functions. Chemical gradients play a decisive role. Molecules called morphogens diffuse through tissues, creating concentration differences. Depending on the amount each cell receives, it interprets a distinct fate. In early embryos, morphogen gradients establish axes, front versus back, head versus tail, left versus right. This spatial information guides development, ensuring that organs appear in the correct places. Signals are not only chemical but also mechanical. Cells pull on one another, rearranging and folding sheets of tissue into layers and organs. The neural tube, precursor of the brain and spinal cord, forms when a flat sheet of cells bends and closes like a zipper. Heart chambers, gut loops, and limb buds all arise from coordinated movements, sculpted by tension and force. Underlying these processes are master regulatory genes. Transcription factors turn on cascades of other genes, steering cells into specific pathways. In fruit flies, for example, homeotic genes determine whether a segment forms a wing, leg, or antenna. In humans, similar genes shape vertebrae, limbs, and organs. Mutations in such genes can lead to dramatic developmental changes, underscoring their central role. Stem cells embody developmental potential. These are undifferentiated cells capable of becoming many types. Embryonic stem cells can form nearly any tissue, while adult stem cells maintain specific organs, replacing worn or damaged cells. In laboratories, scientists study how to direct stem cells to form heart muscle, neurons, or insulin-producing pancreatic cells, opening possibilities for regenerative medicine. To visualize development, imagine an orchestra. At the start, all musicians hold the same sheet of music, yet as the conductor signals, different sections play their parts: strings for melody, brass for harmony, percussion for rhythm. Together they create a symphony far richer than any single instrument. Developmental biology reveals how signals orchestrate cells into tissues and organs, achieving coordinated complexity. Errors in this choreography can have lasting consequences. If signaling pathways misfire, organs may form incorrectly. For example, congenital heart defects arise when chambers or vessels do not develop properly. While treatment lies outside our discussion, the study of such errors informs understanding of both normal development and disease. Advanced imaging now allows researchers to watch embryos develop in real time. Fluorescent markers highlight specific proteins, showing how cells move and divide. Computational models simulate growth, predicting how tissues fold. These tools reveal development not as a rigid plan, but as a dynamic, self-correcting process. Cells communicate continuously, adjusting to ensure that the whole organism emerges with remarkable reliability. A moment of reflection: within you, this silent symphony played long before you were aware. Each organ you now rely upon, heart, brain, lungs, was once a cluster of cells responding to whispers of chemical gradients and mechanical forces. You are the outcome of countless synchronized decisions made by cells without consciousness, yet with precision. This raises the next inquiry. Once an organism has formed, how are its structures organized and described in a way that allows us to comprehend the full architecture of life? When you encounter the human body, or any organism for that matter, you are facing a structure of extraordinary order. Anatomy is the discipline that describes this order. It seeks not only to label organs and tissues, but also to understand how they are arranged in relation to one another. This mapping of form has fascinated observers for millennia. from early dissections in ancient cultures to modern imaging techniques that reveal details without incision. Begin with the simple principle: structure underlies function. A bone has rigidity because its matrix contains mineral deposits. A lung has delicate sacs to maximize surface area for gas exchange. The branching of blood vessels ensures that no cell lies far from oxygen or nutrients. Each part is designed in relation to its role. Anatomy catalogs these forms, creating a guide to the living architecture. The human body can be considered in layers. At the surface lies the integumentary system, skin, hair, nails, and glands, that protects against the environment and regulates interaction with it. Beneath this, muscles provide movement, bones give framework, and joints allow articulation. Deeper still, organs of circulation, respiration, digestion, and excretion interconnect to sustain internal balance. At the core lies the nervous system, coordinating signals, and the endocrine system, orchestrating long-term adjustments. Anatomy presents this not as a random collection, but as an ordered whole where each system relates spatially and functionally to others. To appreciate scale, consider the skeleton. Composed of 206 bones in adults, it provides support, movement, and protection. Each bone has characteristic landmarks, ridges, notches, foramina that serve as attachments or passageways. The skull encases the brain. The vertebral column shields the spinal cord. The ribcage guards the heart and lungs. Bones are not static but living tissues, constantly remodeled by specialized cells that deposit and remove mineral. Their shapes reveal both evolutionary history and mechanical necessity. The study divides into several approaches. Gross anatomy examines structures visible to the naked eye, often through dissection. Microscopic anatomy uses histology, the study of tissues with the microscope, to reveal cellular organization. Developmental anatomy traces how structures arise during growth, while comparative anatomy explores similarities and differences across species. Each perspective contributes to a fuller picture. Now turn to muscles. More than 600 distinct muscles power motion, posture, and expression. Each attaches to bone via tendons, crossing joints to act as levers. Muscles contract when stimulated by nerves, converting chemical energy into mechanical work. Arranged in layers, they overlap and cooperate. Some perform gross movements like extending the leg, while others refine gestures like adjusting the eyes for focus. Muscles reflect both precision and strength depending on their fiber composition and arrangement. The circulatory system illustrates interconnection. The heart, a muscular pump, propels blood through arteries, capillaries, and veins. Arteries branch like rivers, dividing into smaller vessels until reaching capillaries only a single cell thick where exchange occurs. Veins then return the blood, completing the circuit. Within these vessels, red blood cells deliver oxygen while plasma carries nutrients and waste. The spatial layout ensures that every tissue receives continuous support. The digestive tract demonstrates anatomy as a journey. Food enters the mouth, where teeth and saliva begin breakdown. It passes down the esophagus into the stomach, a muscular chamber that churns and acidifies. From there, partially digested material moves to the small intestine, where enzymes and bile complete chemical processing and nutrients are absorbed. The large intestine follows, reclaiming water and housing microbial communities that assist digestion. Finally, waste exits through the rectum and anus. Each segment has structural adaptations suited to its role, from the folds of the stomach to the villi of the small intestine. Anatomy also maps the nervous system, the network that integrates perception and response. The brain, with its lobes and deep nuclei, governs thought, memory, and movement. The spinal cord relays signals between brain and body, with nerves exiting at each level to reach skin and muscle. Peripheral nerves divide into branches, finer than threads, innervating every region. Specialized sense organs, eyes, ears, nose, tongue, skin, convert stimuli into electrical signals, giving awareness of the environment. Even more intricate is the respiratory system. Air enters through the nose or mouth, travels down the trachea, and branches into bronchi and bronchioles, ending in alveoli, tiny sacs surrounded by capillaries. The surface area of alveoli in humans is estimated to be as large as a tennis court, maximizing gas exchange. Their thin walls allow oxygen to diffuse into blood, while carbon dioxide diffuses out. The diaphragm and intercostal muscles control airflow, expanding and compressing the thoracic cavity. Here, anatomy reveals the intimate link between form and survival. One cannot overlook the reproductive systems which illustrate both continuity and variation. In males, testes produce sperm which travel through ducts and mix with secretions from accessory glands to form semen. In females, ovaries release eggs which move through the fallopian tubes where fertilization may occur. The uterus supports development of the embryo, and the external structures provide protection and facilitation of reproduction. Ananopore here bridges individual life with the perpetuation of species. Anatomy of the kidneys demonstrates elegance and filtration. Each kidney contains about 1 million nephrons, microscopic units that filter blood, reclaim useful substances, and secrete waste into urine. Tubules bend and loop to create gradients, ensuring efficient concentration. Positioned in the retroperitoneal space, kidneys connect via ureters to the bladder, which stores urine until excretion. Their structure exemplifies compact design, vast surface area packed into small volume, tuned to balance fluid and electrolytes. To conceptualize anatomy, imagine a map. Landmarks mark organs, roads mark vessels, boundaries mark membranes. The body is a landscape with valleys, ridges, and rivers, all arranged for function. Like a cartographer, the anatomist charts this terrain, naming each feature and noting its connections. This mental map allows navigation by clinicians, whether placing a stethoscope, interpreting an image, or planning a surgical approach. Errors in anatomy can have profound consequences. A congenital malformation such as transposition of the great vessels alters the path of blood flow, requiring recognition and correction. Variations in branching of arteries may complicate surgery. Tumors may compress nearby structures with symptoms depending on anatomical relationships. Thus, knowledge of anatomy is not abstract but essential for safe practice. Modern technology deepens anatomical study. Imaging modalities such as computed tomography (CT) and magnetic resonance imaging (MRI) generate detailed cross-sections of the body. Ultrasound provides real-time views of moving organs. Three-dimensional reconstructions allow visualization from angles impossible with dissection. These methods extend anatomy beyond cadaver study, showing living structures in action. A clinical case illustrates why layered knowledge matters. Consider appendicitis. Pain often begins diffusely near the navel, reflecting nerve supply to the gut. As inflammation progresses, pain localizes to the right lower quadrant where the appendix lies. Knowing the anatomical pathway of pain explains this shift. Surgeons rely on precise maps of blood supply and neighboring organs to remove the appendix safely. Anatomy thus guides both diagnosis and intervention. Histology adds another dimension. Under the microscope, tissues reveal cellular arrangements that support organ function. Epithelial cells line surfaces, forming barriers and glands. Connective tissue provides scaffolding from dense collagen in tendons to elastic fibers in lungs. Muscle tissue appears in 3 varieties: skeletal, smooth, and cardiac, each suited to its task. Nervous tissue displays neurons with long extensions and supporting glial cells. Together, these tissues form the microscopic basis of gross anatomy. Reflection here becomes almost inevitable. When you close your eyes and breathe, you may sense only the rise of your chest, yet beneath lies a cathedral of structures: bones arching, muscles pulling, airways branching, blood coursing, neurons firing. Anatomy invites you to envision this cathedral in detail, to appreciate the architecture that makes your life possible. The wonder leads naturally to the next question. If anatomy charts what organisms are made of, then how do these structures actually perform their tasks, and how do their functions interlock into the living whole? Movement is one of the most visible expressions of life. When you walk, lift, breathe, or even blink, an immense interplay of mechanical forces unfolds within your body. The study of these forces belongs to biomechanics, which seeks to explain how structures are engineered for motion and resilience. Unlike anatomy, which maps what is present, biomechanics interprets how those structures behave under load, tension, compression, or flow. Begin with the skeleton, the rigid framework. Each bone is not merely a support beam, but also a lever. When muscles contract, they pull on tendons, which transmit force to bones, causing rotation at joints. This lever system amplifies movement. In your arm, for instance, the biceps attaches close to the elbow joint. Though this short lever requires more muscular force, it allows rapid and precise movement of the hand. Evolution favors such trade-offs, sometimes strength, sometimes speed, sometimes fine control. The joints themselves are marvels of engineering. Synovial joints, the most mobile type, contain cartilage that cushions, synovial fluid that lubricates, and ligaments that stabilize. Their shapes, hinge, ball and socket, pivot, determine the possible motions. A hinge joint like the knee allows flexion and extension, but little else. A ball and socket joint like the hip permits rotation in many directions. Biomechanics analyzes how these forms distribute forces, resist wear, and adapt to stress. Muscles add power. Skeletal muscle fibers contract when myosin filaments slide past actin filaments powered by ATP. The arrangement of fibers influences performance. Pinnate muscles with fibers angled to tendons pack more fibers in limited space, providing strength. Parallel muscles with fibers aligned offer greater range of motion. Thus, the calf muscle emphasizes power, while the sartorius in the thigh emphasizes length. The heart demonstrates biomechanics at a different scale. Each beat depends on coordinated contractions of cardiac muscle, generating pressure that drives blood through vessels. Valves prevent backflow, opening and closing in response to pressure gradients. The physics of fluid flow applies. Resistance, velocity, turbulence. Arteries expand with each pulse, then recoil, smoothing flow. Veins rely on valves and surrounding muscle contractions to return blood to the heart. Here biomechanics explains how motion of fluid sustains life. At the microscopic level, cells themselves have mechanical properties. Their membranes flex, their cytoskeletons provide scaffolding, and they transmit forces to their environment. White blood cells squeeze through capillaries narrower than themselves, deforming yet recovering. Bone cells sense mechanical load and respond by remodeling bone, strengthening regions under stress. Thus, biomechanics extends from whole limbs to single cells. Consider breathing. The diaphragm contracts, lowering the floor of the thoracic cavity, while external intercostal muscles lift the ribs. These changes expand the lungs, drawing in air. Exhalation, at rest, is largely passive as muscles relax and elastic tissues recoil. The mechanics follow pressure gradients. Air moves from higher to lower pressure, yet during exercise or illness, accessory muscles add force, demonstrating adaptability of the system. Biomechanics also interprets movement in other species, offering comparative insight. Birds have hollow bones to reduce weight, wings shaped for lift, and muscles adapted for sustained contraction. Fish propel themselves with waves of muscle contractions, transmitted to fins, exploiting the density of water. Cheetahs use flexible spines as springs, extending stride length for rapid acceleration. Each adaptation reflects optimization of mechanical principles. To picture biomechanics, imagine bridges and cranes. Engineers calculate stresses, bending moments, and load distributions to ensure stability. In the body, natural selection has achieved similar engineering solutions. Bones arch like girders, muscles act as cables, joints as hinges, fluids as hydraulic systems. The entire organism functions as a dynamic structure under constant load, adapting continuously. Clinical relevance is clear. In orthopedics, understanding biomechanics guides prosthetic design, joint replacement, and fracture repair. Artificial hips, for instance, must replicate load distribution to prevent loosening. In cardiology, biomechanics informs stent design, ensuring blood flow remains smooth. In rehabilitation, therapists apply biomechanical principles to restore gait, posture, or hand function. When mechanics fail, such as ligament tears, valve defects, or vessel aneurysms, understanding forces clarifies both symptoms and risks. Reflect for a moment. Every step you take depends on countless silent calculations of force, angle, and resistance. Your body is both machine and organism, solving problems of physics with elegance. This reflection invites the next inquiry. If biomechanics explains how structures move, then how do those structures across every organ system cooperate in real time to sustain life itself? To understand the body as more than assembled parts, you must shift from structure to activity. Physiology studies how organs and systems perform their tasks and how their functions weave into a coherent whole. If anatomy is the map, Physiology is the journey, showing how traffic flows, how signals cross, how balances are maintained in constant dialogue. Begin with the principle of homeostasis. This term refers to the body's ability to maintain stable internal conditions despite external change. Temperature, pH, glucose concentration, oxygen levels, all must remain within narrow ranges. Sensors detect deviations, control centers interpret signals, and effectors restore balance. This feedback system resembles a thermostat. When a room cools, the heater activates. When it warms, the heater stops. In the body, countless thermostats operate simultaneously, invisible yet essential. Consider temperature regulation. When heat rises, blood vessels in the skin dilate, and sweat glands secrete fluid that evaporates, cooling the body. When cold prevails, vessels constrict, conserving heat, and muscles shiver to generate warmth. These responses involve coordination between nervous and endocrine systems, between circulatory and muscular systems, between individual cells and the whole organism. Physiology explains this coordination as the harmony of feedback loops. The cardiovascular system offers another window. The heart pumps rhythmically, establishing blood pressure that propels fluid through vessels. Baroreceptors in arteries monitor pressure, sending signals to the brainstem. If pressure drops, sympathetic nerves increase heart rate and constrict vessels. If it rises, parasympathetic signals slow the heart and dilate vessels. This constant adjustment ensures that tissues receive sufficient blood supply, whether you rest or run. Without such control, life would collapse in moments. Respiration demonstrates integration across levels. Oxygen enters lungs, diffuses into blood, binds hemoglobin, and travels to tissues. Carbon dioxide moves in the opposite direction. Sensors in the brainstem and arteries monitor carbon dioxide, not oxygen, as the primary driver of breathing. When levels rise, signals intensify to breathe faster and deeper. Muscles expand the thorax, air flows in, and balance is restored. The rhythm persists even during sleep, governed by automatic centers but adaptable when you speak, sing, or dive underwater. The digestive system reveals physiology as sequence. Food broken down mechanically in the mouth is chemically processed by enzymes and acids in the stomach and intestines. Hormones regulate timing, gastrin stimulates acid release, secretin signals pancreatic secretions, cholecystokinin triggers bile, peristalsis, rhythmic waves of muscle contraction, moves contents forward. Meanwhile, absorption occurs through specialized epithelial cells. Thus, physiology shows how digestion is not random but orchestrated, each step prepared for the next. The kidneys embody homeostasis in miniature. They filter blood, reclaim water, adjust electrolytes, and excrete waste. Nephrons act as functional units. Blood enters glomeruli where filtration occurs. Tubules reabsorb useful substances. Loops create gradients for water reabsorption. Collecting ducts adjust final concentration. Hormones like aldosterone and antidiuretic hormone fine-tune the process. The result is precise control of volume and composition, essential for survival. Hormonal regulation illustrates physiology at a systemic level. The endocrine system releases chemical messengers into blood, reaching distant targets. The hypothalamus in the brain monitors internal state, signaling the pituitary gland, which then influences thyroid, adrenal, and gonadal glands. Cortisol prepares the body for stress. Thyroid hormones adjust metabolic rate. Insulin and glucagon regulate glucose. These hormones act slowly compared to nerves, but sustain long-term balance. Nervous control, by contrast, acts rapidly. Neurons transmit electrical impulses, releasing neurotransmitters across synapses. Reflex arcs demonstrate simplicity. Touch a hot surface, and sensors in skin send signals to spinal cord, which immediately directs muscles to withdraw, often before you consciously perceive pain. Yet the same system underlies thought, memory, and emotion, integrating physiology with mind. You can imagine physiology as a vast conversation. Organs are speakers, hormones and nerves are words, and feedback loops are grammar ensuring meaning. The dialogue never ceases. Silence would equal death. The wonder lies in its adaptability. Whether fasting or feasting, sleeping or exercising, at sea level or high altitude, the body adjusts through physiological mechanisms. Comparative physiology highlights diversity of solutions. Desert animals concentrate urine to conserve water, while aquatic creatures excrete dilute urine freely. Birds possess unidirectional airflow through lungs, extracting oxygen more efficiently than mammals. Diving mammals slow heart rate and redirect blood to essential organs, enduring long periods without breathing. These variations reveal evolution's ingenuity, sculpting physiology to meet environmental demands. Clinical contexts reveal consequences when physiology falters. In heart failure, pumping weakens, pressure control mechanisms strain, and tissues starve for oxygen. In diabetes, insulin regulation of glucose fails, leading to high blood sugar and metabolic imbalance. In chronic lung disease, impaired gas exchange drives increased breathing effort, altering cardiovascular load. Each condition reflects disruption of feedback loops once seamless. Recognition of these disruptions allows clinicians to interpret symptoms, even though treatment lies beyond our scope. To grasp the scale, consider exercise. Muscles contract repeatedly, demanding more oxygen and energy. The heart increases output, vessels dilate in active regions, breathing deepens, liver releases glucose, sweat glands activate. This coordinated response involves virtually every system. When exercise stops, signals reverse, returning the body to rest. Physiology captures this dance of demand and recovery, a demonstration of resilience. Technology now illuminates physiology in real time. Imaging shows blood flow and beating hearts, sensors record neural activity, tracers track hormones through circulation, computer models simulate complex interactions, predicting responses to interventions. These tools transform physiology from hidden processes into observable dynamics, reinforcing its role as the science of living function. Reflection may come gently, though you rest quietly. Within you, thousands of adjustments unfold each second. Pressure corrected, temperature balanced, glucose managed, oxygen delivered. You are not a passive structure but an active equilibrium, sustained through invisible labor. The inevitable question follows: if the body defends its balance so carefully, what specialized system exists to protect against threats not from within but from the outside world? Your body is constantly exposed to threats: bacteria, viruses, fungi, parasites, and even malfunctioning cells of its own. Yet most of the time you remain healthy, unaware of the microscopic battles being fought within you. The system responsible for this vigilance is the immune system, a vast network of cells, tissues, and molecules that recognizes danger, responds, and restores balance. Immunology studies this system in detail, uncovering how protection is mounted and why it sometimes misfires. Begin with the two broad divisions, innate and adaptive immunity. The innate system is ancient, present in nearly all multicellular organisms. It acts quickly, responding within minutes or hours. Skin and mucous membranes provide barriers, while specialized cells such as macrophages and neutrophils engulf invaders. Chemical defenses include antimicrobial peptides and the complement system, a cascade of proteins that puncture microbial membranes. Innate immunity recognizes patterns common to many pathogens like bacterial cell walls, allowing broad but nonspecific protection. Adaptive immunity, unique to vertebrates, is slower to respond initially but highly specific and capable of memory. Lymphocytes, B cells and T cells form its core. B cells produce antibodies, proteins that bind to particular antigens with exquisite precision. T cells come in several types. Helper T cells coordinate responses, cytotoxic T cells kill infected cells, and regulatory T cells restrain excessive activity. Once adaptive immunity encounters a pathogen, it remembers. On re-exposure, the response is faster and stronger, forming the basis for long-lasting protection. Consider the timeline of a respiratory infection. A virus enters through inhalation, reaching the airway epithelium. Innate defenses respond first. Mucus traps particles, cilia sweep them upward, interferons signal nearby cells to mount antiviral states. If the virus persists, antigen-presenting cells capture fragments and travel to lymph nodes, where they display them to T cells. Recognition triggers clonal expansion, proliferation of specific lymphocytes armed against that pathogen. B cells begin secreting antibodies that neutralize viral particles, while cytotoxic T cells patrol for infected host cells to destroy. Once the infection clears, most effector cells die, but memory cells remain, ready for future encounters. The cooperation between systems is intricate. Antibodies coat bacteria, marking them for phagocytosis. Complement proteins enhance this marking and recruit immune cells. Cytokines, small signaling molecules, create communication networks that amplify or suppress responses. Too little response leaves the body vulnerable. Too much risks collateral damage to tissues. Balance is the constant goal. To picture the immune system, imagine a city's defense. Walls and guards at the gates resemble barriers and innate cells. Specialized detectives search for criminals with unique signatures like T and B cells recognizing antigens. Once identified, alarms spread, reinforcements arrive, and targeted strikes remove threats. Yet after the crisis, forces withdraw, leaving behind intelligence archives, memory cells that prepare the city for similar attacks. Failures of immunity teach as much as successes. When the system underreacts, infections flourish. People with impaired immune responses may struggle against everyday microbes. When the system overreacts, allergies emerge. Harmless substances like pollen or food proteins trigger excessive responses, causing sneezing, swelling, or even life-threatening anaphylaxis. In autoimmunity, the system mistakenly targets self, leading to conditions such as type 1 diabetes, rheumatoid arthritis, or multiple sclerosis. In cancer, malignant cells sometimes escape recognition by disguising themselves or suppressing immune activity. Each of these illustrates the delicate balance immunity must achieve. Clinical practice relies heavily on immunological principles. Vaccination exemplifies applied immunology. By exposing the body to harmless components of a pathogen, the adaptive system generates memory without full disease. The next time the real pathogen appears, the immune response is swift and potent. Beyond prevention, immunology informs diagnostics. The presence of specific antibodies can indicate prior exposure, while counts of immune cells reveal states of suppression or activation. Even therapies in oncology increasingly exploit immunity, using engineered T cells or checkpoint inhibitors to unleash attacks against tumors. The tools used to study immunity are equally impressive. Flow cytometry sorts thousands of cells per second, identifying types based on surface markers. Microscopy with fluorescent labels shows cells migrating, engaging, and killing targets. Genomic methods sequence antibody repertoires, revealing the diversity of recognition. Each tool expands understanding, yet also underscores complexity. The immune system is not one entity but a constellation of processes, cooperating and competing, guided by both chance and regulation. Comparative immunology shows how different organisms solve the same problem. Insects rely on innate mechanisms only, yet survive effectively. Jawless fish, among the most ancient vertebrates, use unique molecules instead of antibodies to achieve specificity. Mammals combine speed of innate responses with precision of adaptive memory. These comparisons highlight evolution's creativity, shaping defenses to match environmental pressures. Reflecting on your own life, you rarely notice the immune system until it falters. A fever signals its activity, inflammation marks its presence, fatigue accompanies its effort, yet most of the time it operates silently, a hidden guardian. Every cut you have healed, every infection you have overcome, reflects its constant vigilance. But vigilance has a cost. A question arises: how has human health improved so dramatically in recent centuries? And what role has scientific study of biology played in prolonging life? The length of human life has expanded dramatically in just a few centuries. Where once infectious disease and injury often cut existence short, today many people live decades longer, reaching ages unimaginable to their ancestors. This transformation is not the result of chance. It is the outcome of systematic investigation into how the body functions, what harms it, and how to intervene. Biomedical research stands at the heart of this achievement, uniting basic biological knowledge with applied discovery to confront illness and extend survival. The journey begins with observation. When a new illness arises, scientists study its clinical signs, its transmission, and its biological footprint. From these observations, hypotheses emerge about underlying mechanisms. Investigators then design experiments, often starting at the molecular or cellular level. In laboratories, cell cultures reveal how viruses invade, how toxins damage, or how signals fail. Animal models extend this, demonstrating how whole organisms respond. Each layer of study provides a fragment of the puzzle. Biomedical research weaves these fragments into explanations that eventually translate into prevention, diagnosis, or therapy. At the most fundamental level, biomedical research examines molecular pathways that sustain life and how these pathways break in disease. Consider cancer. At its core, cancer is uncontrolled cell growth. By studying signaling pathways, scientists uncover mutations that disable checkpoints, activate growth signals, or block programmed cell death. These insights explain why tumors form and guide strategies to detect them earlier. Or take Alzheimer's disease. By analyzing protein accumulation in neurons, researchers explore how misfolded molecules disrupt communication and lead to decline. Each discovery clarifies not only one illness, but principles that ripple across biology. Diagnosis advances hand in hand. When researchers identify markers unique to disease, clinicians gain tools to detect illness earlier and with greater precision. Biomarkers may be molecules in blood, genetic variations, or imaging signatures. For example, elevated troponin levels in blood provide early evidence of heart attack, even before symptoms fully manifest. Tumor markers suggest cancer recurrence, while genetic screens reveal predispositions to inherited disorders. Without basic research to identify these markers, such tests would not exist. Technology serves as both tool and outcome of research. The microscope once opened the world of cells. Today, sequencing machines open the world of genomes. Imaging technologies such as MRI and PET scans emerged from research into physics and physiology, yet now serve daily in clinics. Miniaturized sensors track heart rhythms continuously, implantable devices stimulate nerves, and robotic systems assist surgery. Each innovation reflects decades of investigation into how biological systems can be monitored, guided, or supported by devices. Pharmaceutical development illustrates the path from concept to clinic. It begins with discovery, identifying molecules that interact with biological targets. High-throughput screening allows testing of thousands of compounds against proteins implicated in disease. Promising candidates undergo refinement, ensuring they bind strongly, act specifically, and remain stable in the body. Preclinical studies test safety and efficacy in models. Only then do clinical trials begin, moving through phases, first assessing safety in small groups, then evaluating effectiveness, then confirming results across large populations. Each stage narrows the field but increases confidence that the final product will help rather than harm. One metaphor clarifies this process. Imagine building a bridge across a wide river. Theoretical designs on paper resemble basic discoveries in the lab. Scale models tested in controlled conditions are like preclinical experiments. Only after repeated testing and adjustments does the full bridge rise, sturdy enough for daily use. In biomedical research, the bridge spans from molecular insight to patient care, each stage essential, each failure instructive. Yet, biomedical research is not confined to drugs. Devices also change the landscape. Artificial joints restore mobility, pacemakers regulate heart rhythm, and prosthetics integrated with neural signals restore partial control of limbs. Even artificial organs such as dialysis machines replacing kidney function extend life while awaiting transplant. Behind each lies decades of incremental refinement, guided by biological knowledge and engineering ingenuity. Synthetic biology now expands possibilities further. By designing organisms with altered genetic codes, researchers create bacteria that produce insulin, yeasts that generate vaccines, or cells engineered to attack tumors. These applications illustrate how biology itself can be harnessed as a tool, not merely studied as an object. The boundary between research and engineering blurs, with life reimagined as both subject and instrument of discovery. But biomedical research also faces limits and responsibilities. Safety remains paramount. History records tragedies when interventions rushed to use without adequate testing caused harm. Modern regulations enforce rigorous oversight. Ethical review boards, informed consent, phase testing, and continuous monitoring. Privacy concerns also emerge as genetic data accumulate, requiring careful stewardship. The power of biomedical research demands humility, caution, and constant evaluation. Reflect on pandemics to see its urgency. When a novel virus appears, research mobilizes at unprecedented speed. Sequencing its genome, developing diagnostic tests, screening treatments, designing vaccines. International collaboration accelerates progress, but challenges of distribution, access, and public trust remain. Research alone cannot solve crises. Translation into policy and practice must follow. Still, without the scientific foundation built over decades, responses would be blind and slow. A clinical example shows how research reshapes possibility. Before the discovery of antibiotics, infections that today are trivial could be fatal. Simple wounds, childbirth, or dental procedures often led to sepsis. Biomedical research into microbial physiology revealed how certain compounds block bacterial processes, leading to the development of penicillin and subsequent antibiotics. Lifespan lengthened not only because infections were cured, but because surgery, transplants, and chemotherapy, all dependent on infection control, became feasible. Similarly, vaccines demonstrate cumulative progress. By studying viral structure and immune responses, researchers design antigens that safely train the immune system. Diseases once widespread, such as smallpox or polio, have been eradicated or nearly eliminated in many regions. This represents not only scientific triumph but also collaboration between research, policy, and public health. The scale of biomedical research spans from rare disorders affecting a few families to global diseases threatening millions. Rare disease research often uncovers fundamental principles, since single gene defects reveal essential pathways. For example, study of hemophilia, a disorder of clotting, clarified mechanisms of coagulation relevant to all patients. Thus, even focused investigations ripple outward, broadening knowledge across medicine. Modern biomedical research integrates multiple disciplines. Genomics deciphers DNA, proteomics catalogs proteins, systems biology maps interactions into networks, computational biology simulates processes, guiding experiments, engineering designs tools, while chemistry creates compounds. The convergence of these fields accelerates discovery, creating synergy greater than the sum of parts. A metaphor captures this integration. Imagine a vast orchestra where each instrument represents a discipline. Alone, a violin or a drum contributes a fragment. Together, under careful coordination, they produce symphonies of understanding. Biomedical research is the conductor, weaving insights into coherent outcomes that touch human lives. Clinical translation remains the ultimate measure. A laboratory finding, however elegant, achieves full meaning only when it alters health outcomes. This translation is complex, requiring not only scientific validation but also cost-effectiveness, accessibility, and acceptance. An expensive drug inaccessible to most patients or a device too fragile for everyday use cannot fulfill its promise. Thus, researchers, clinicians, economists, and policymakers collaborate to bridge discovery and delivery. Pause for reflection. Behind every pill in a cabinet, every image on a screen, every vaccination scar on an arm, lies decades of unseen work. Teams of scientists, clinicians, and patients contributed fragments of knowledge, stitched together into interventions. The lengthened lifespan you enjoy is not accidental, but crafted through persistent inquiry. And yet, despite such progress, questions multiply. Why do some diseases remain incurable? How far can biology be engineered without crossing ethical boundaries? How can discoveries be shared equitably across nations and populations? These questions remind you that biomedical research is both an achievement and a responsibility, a testament to human curiosity, but also a call for prudence. You have seen how science prolongs life and alleviates suffering. But as research merges with engineering, another question arises. Can we not only repair life but redesign it, creating entirely new forms that never existed before? Life can be repaired, supported, and extended, but biomedical research eventually invited a deeper question: can life itself be redesigned? Out of this question emerged the field of bioengineering, where principles of engineering meet the knowledge of biology, and from their intersection arise devices, therapies, and even synthetic organisms. This discipline does not stop at describing the body, it seeks to intervene, to improve, and in some cases to create anew. The story begins with prosthetics, the earliest attempts to replace lost function with artificial tools. Wooden limbs, simple hooks, or carved teeth stand as ancient examples, but modern bioengineering transforms these into sophisticated devices. Prosthetic limbs now integrate lightweight alloys, microprocessors, and neural interfaces. By detecting signals from residual muscles or even directly from nerves, they allow intuitive control of artificial hands that can grasp, rotate, and sense pressure. Each advance reflects not just craftsmanship but systematic study of biomechanics, neurology, and material science. Artificial organs demonstrate the next step. Dialysis machines replace kidney function by filtering blood outside the body, removing waste and balancing electrolytes. Ventricular assist devices help failing hearts pump blood. Artificial corneas restore vision, while cochlear implants translate sound into electrical signals interpretable by the auditory nerve. In each case, engineers and biologists collaborate, mimicking natural processes with mechanical or electronic counterparts. These devices reveal that life can be sustained even when original organs falter. But bioengineering extends beyond substitution. In tissue engineering, scaffolds made of biocompatible materials provide frameworks for cells to grow, forming cartilage, skin, or even segments of blood vessels. Researchers culture stem cells on these scaffolds, coaxing them into organized tissues. One vision is to grow entire organs in the laboratory, using a patient's own cells to avoid rejection. Although still experimental, progress already shows patches of tissue repairing damaged hearts or bioengineered skin aiding burn victims. The principle is simple but powerful: provide cells with structure, signals, and support, and they can rebuild what was lost. Synthetic biology expands this ambition further. By rewriting genetic codes, scientists design organisms with new capabilities. Bacteria are engineered to produce insulin, an achievement that replaced extraction from animal pancreases and revolutionized diabetes management. Yeast cells are modified to manufacture vaccines or rare drugs. Even microorganisms are designed to sense toxins, degrade pollutants, or generate biofuels. These creations do not merely imitate natural functions, they extend them, crafting life as a tool for medicine, industry, and environment. One striking illustration is the use of engineered immune cells. In a therapy known as KRT, a patient's own T lymphocytes are modified to express receptors targeting cancer cells. Reintroduced into the body, these cells seek out and destroy malignancies that previously resisted treatment. Though not without risks, the success of such therapies demonstrates how bioengineering redefines the boundaries of possibility. Cells themselves become living medicines. The metaphor of architecture helps here. Biology provides bricks, beams, and mortar cells, proteins, and DNA. Engineering supplies blueprints, cranes, and scaffolds. Together they construct not only replacements for damaged buildings but entirely new structures. A bacterium producing a pharmaceutical compound is a custom-designed factory. A synthetic circuit of genes is a programmed machine, built not from silicon but from nucleotides. Ethical considerations rise in parallel. To redesign life is to wield profound power. Concerns include unintended consequences, ecological disruption if engineered organisms escape, inequities in access, and questions about the definition of life itself. Regulatory frameworks attempt to balance innovation with caution, requiring safety testing, containment measures, and oversight. Bioengineering thus carries both promise and responsibility, demanding humility as well as creativity. Clinical implications are immediate. Patients with organ failure once faced limited options, transplantation with all its challenges of supply and rejection or progressive decline. Now bioengineered alternatives suggest new futures. Artificial pancreases integrate glucose sensors with insulin pumps, automating control for diabetes. Neural prosthetics restore some movement to paralyzed individuals by bridging damaged pathways. Even brain-computer interfaces are under exploration, where thought alone may guide machines. Each example demonstrates the union of knowledge and engineering. Comparative approaches enrich the field. Nature itself is a master engineer. Spider silk surpasses steel in strength-to-weight ratio. Lotus leaves repel water through microscopic surface patterns. Electric eels generate bioelectricity. By studying these adaptations, engineers design materials and devices inspired by biology, a practice known as biomimicry. From adhesives modeled on gecko feet to lenses mimicking insect eyes, the living world supplies templates for innovation. Synthetic biology's most daring frontier is the creation of genomes from scratch. researchers have synthesized entire bacterial chromosomes and inserted them into cells, producing organisms guided by human-written DNA. Though still rudimentary, this achievement suggests the possibility of designing life forms entirely new to Earth. Applications may include microbes optimized for medicine, agriculture, or industry. Yet such advances also stir debate. Should life be considered an invention, and if so, what responsibilities accompany its creation? Bioengineering also extends into environmental realms. Engineered algae produce biofuels, potentially reducing reliance on fossil energy. Modified plants resist drought or pests, aiming to secure food supplies under changing climates. Microbes tailored to degrade plastics or capture carbon dioxide promise mitigation of pollution and climate change. Thus, the reach of bioengineering spans not only health but global ecosystems. Imagine for a moment that life itself becomes a toolkit. DNA sequences are instructions, proteins are components, and cells are factories. Engineers rearrange these parts to produce outcomes once thought impossible. The power is extraordinary, yet the task is to wield it wisely, ensuring that interventions heal rather than harm. From a clinical perspective, it is worth noting how close bioengineering is to everyday life. A pacemaker implanted in a chest, a prosthetic knee enabling walking, a genetically engineered insulin vial on a pharmacy shelf, these are not futuristic but present. They remind you that bioengineering is not abstract but tangible, woven into the very fabric of modern health. Reflection arises naturally. You are the inheritor of a world where biology can be not only understood but redesigned. What once required natural selection over millions of years may now be achieved in laboratories within decades. Yet each success carries questions about boundaries and stewardship. From this vantage point, another inquiry follows. If we can re-engineer cells and organs, how does the most complex system of all, the brain, function as both a biological machine and the seat of thought? The nervous system is the most intricate and enigmatic of all biological architectures. It is at once a network of cells transmitting electrochemical impulses and the seat of sensation, memory, and awareness. To study it is to stand at the intersection of biology and philosophy, for within its circuits arise not only reflexes and movements, but also perception and thought. Neuroscience seeks to unravel this complexity, explaining how neurons, glia, molecules, and circuits converge into a functioning whole. Begin with the fundamental unit, the neuron. Each neuron consists of a cell body, dendrites that receive signals, and an axon that transmits them. At the end of the axon, synaptic terminals release neurotransmitters into narrow clefts, bridging to the next cell. This chain of communication occurs with astonishing speed, milliseconds between perception and response. Electrical impulses arise from the movement of ions across membranes through specialized channels, creating action potentials that propagate like waves down axons. The fidelity of this system ensures precise control, whether moving a finger or recalling a memory. Supporting neurons are glial cells, once thought to play minor roles but now recognized as essential. Astrocytes regulate neurotransmitter levels and supply metabolic support. Oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system wrap axons with myelin, insulating them and increasing conduction velocity. Microglia act as immune sentinels, patrolling the brain for injury or infection. Together, glia provide the environment in which neurons thrive, illustrating that even the most celebrated cell relies on collaboration. From single cells arise circuits. Reflex arcs demonstrate simplicity. Sensory neurons detect stimuli, interneurons process information, and motor neurons elicit response. More complex circuits govern locomotion, breathing, or sleep. In the brain, vast networks coordinate to integrate sensory input, store information, and plan actions. These circuits adapt through synaptic plasticity, the ability of connections to strengthen or weaken based on activity. Plasticity underlies learning and memory, allowing experiences to sculpt neural architecture. At higher levels, the nervous system organizes into distinct regions. The spinal cord acts as a conduit and processor for reflexes. The brainstem controls vital functions such as breathing, heart rate, and arousal. The cerebellum refines motor activity, ensuring coordination and balance. The diencephalon houses the thalamus, relaying sensory signals, and the hypothalamus, orchestrating endocrine and autonomic regulation. Above these lies the cerebral cortex, a folded sheet of neurons responsible for perception, language, reasoning, and voluntary movement. Divided into lobes, frontal, parietal, temporal, and occipital, the cortex reflects specialization within unity. Consider vision. Light enters the eye, focused onto the retina, where photoreceptors convert photons into electrical signals. These signals pass through layers of neurons, converge on ganglion cells, and travel via the optic nerve to the brain. There, the thalamus relays them to the occipital cortex, where edges, colors, and motion are analyzed. From there, signals integrate with memory and emotion, allowing recognition, a face, a landscape, a word. Vision exemplifies how sensory input becomes experience, shaped by both external stimuli and internal interpretation. Language provides another illustration. Broca's area in the frontal lobe governs speech production, while Wernicke's area in the temporal lobe manages comprehension. Damage to one disrupts expression, to the other comprehension. Yet both function only within broader networks connecting auditory, visual, and motor regions. Thus, even tasks we consider singular emerge from distributed cooperation across circuits. The metaphor of an orchestra applies again, but at a new scale. Neurons are instruments, glia are tuners, neurotransmitters are notes, and brain regions are sections of the ensemble. The symphony is not static but constantly improvising, modulating tempo, tone, and harmony. Consciousness itself may be the emergent music of this orchestra, arising not from one instrument, but from the coordinated performance of all. Electrical activity can be observed through techniques like electroencephalography, EEG, which records brain waves, or functional magnetic resonance imaging, fMRI, which tracks blood flow as a proxy for activity. These tools reveal patterns during sleep, attention, or decision-making. They show that brains never truly rest, even in quiet states. Networks hum with intrinsic activity, maintaining readiness and integrating prior experiences. Neuroscience also explores pathology to illuminate function. In epilepsy, sudden bursts of synchronized activity disrupt normal patterns, causing seizures. Studying these events reveals mechanisms of excitability and inhibition. In Parkinson's disease, degeneration of dopamine-producing neurons in the midbrain leads to tremor and rigidity, highlighting dopamine's role in movement. In depression, altered neurotransmitter dynamics and circuit connectivity suggest mechanisms of mood regulation. Each disorder not only challenges health but also teaches about the normal functions now disrupted. A clinical vignette shows the stakes. A patient suffering a stroke loses blood supply to part of the brain. Depending on the region affected, deficits may include paralysis, speech loss, or memory impairment. Yet over time, other circuits may adapt, restoring partial function. This recovery illustrates neuroplasticity, the ability of the brain to rewire and compensate. Rehabilitation strategies harness this plasticity, guiding the brain's own capacity to heal. Advances push boundaries further. Neuroprosthetics link brain activity to external devices, allowing paralyzed individuals to control robotic arms through thought. Brain-computer interfaces decode patterns of activity, translating intention into action. These achievements highlight both the power and mystery of the nervous system. If signals can be read and translated, then thought itself becomes a measurable, utilizable process. Comparative neuroscience expands perspective. Octopuses with distributed nerve networks in their arms demonstrate decentralized intelligence. Birds, despite small brains, perform complex tasks and migrations, challenging assumptions about brain size and cognition. Insects navigate, communicate, and coordinate in swarms with tiny nervous systems. These examples remind us that intelligence takes many forms, shaped by ecological demands and evolutionary history. Your every thought, memory, and feeling arises from patterns of electrochemical activity. That you can reflect on your brain means the brain has turned its capacity inward, examining itself. Few phenomena are as paradoxical, matter arranged to contemplate its own arrangement. And yet the nervous system does not act alone. To change mood, perception, or response, chemical substances, whether produced internally or administered externally, interact with it. This leads to the next inquiry: how do drugs and disease processes alter biological systems, and what do these changes reveal about the delicate balance of life? The effects of chemical substances on living systems have fascinated and troubled humanity since antiquity. Some plants soothed pain, others poisoned, some extracts lifted mood, others clouded thought. Over centuries, systematic study transformed this trial and error knowledge into pharmacology, the science of how drugs interact with the body, and pathology, the science of how diseases alter it. Together they reveal how biology can be shifted off balance and how it can sometimes be steered back toward health. Begin with pharmacology. At its heart lies the concept of the drug-receptor interaction. Cells are studded with proteins, receptors that receive signals. A neurotransmitter binds its receptor to open an ion channel. A hormone binds another to alter gene expression. Drugs exploit these same targets, sometimes mimicking natural signals, sometimes blocking them. An analgesic may prevent pain signals from being transmitted. An antihistamine may block receptors that mediate allergic responses. The principle is specificity. A single molecule, by binding to a particular receptor, can alter a cascade of processes. But the body is not passive. Every drug, once taken in, must be absorbed, distributed, metabolized, and excreted. Pharmacokinetics studies this journey. Absorption determines how a drug enters the bloodstream through the gut, lungs, skin, or injection. Distribution depends on blood flow and solubility, deciding which tissues the drug reaches. Metabolism, primarily in the liver, transforms molecules into more water-soluble forms for elimination. Excretion through kidneys, bile, or lungs finally removes them. Each step affects how long and how strongly a drug acts. Pharmacodynamics, in contrast, asks what the drug does to the body. Does it activate a receptor, an agonist, or block it, an antagonist? Does it enhance a pathway or inhibit it? Effects may be therapeutic, but they may also be adverse. A sedative may calm but also impair coordination. An anti-inflammatory may reduce pain but irritate the stomach. This duality is inherent. Every powerful action risks unintended consequence. The metaphor of a key and lock helps. A drug is a key, a receptor, a lock. Some keys fit perfectly, turning the lock to open pathways. Others jam the lock, preventing natural keys from entering. Yet not all locks are isolated. Many are part of larger doors and mechanisms. A drug that opens one door may inadvertently affect another. Pharmacology seeks to understand these connections, predicting benefit and risk. Pathology enters when balance fails not by external intervention but by internal disruption. It studies structural and functional changes in tissues and cells that underlie disease. Inflammation is one example, a protective response to injury or infection characterized by redness, swelling, heat, pain, and loss of function. Microscopically, blood vessels dilate, immune cells migrate, and mediators flood the area. When controlled, this process removes harmful agents and initiates repair. When excessive or chronic, it damages tissue, leading to conditions such as arthritis. Cell injury and adaptation form another focus. Cells exposed to stress may hypertrophy, grow larger, hyperplasia, increase in number, atrophy, shrink, or undergo metaplasia, change type. These adaptations may preserve function temporarily, but can become pathological if prolonged. Necrosis describes uncontrolled cell death, often with inflammation, while apoptosis is programmed, orderly cell death essential for development and homeostasis. Cancer arises when growth escapes regulation, cells divide uncontrollably, and tissues invade others. Pathology categorizes these changes, providing insight into both cause and effect. In practice, pathology relies heavily on samples, blood smears, biopsies, autopsies. A tumor examined under a microscope may reveal its type, grade, and likely behavior. An inflamed organ may show whether infection or autoimmunity drives the process. Post-mortem examination not only clarifies cause of death, but also advances collective knowledge, identifying new diseases or complications. Thus, pathology bridges observation at the bedside with understanding at the cellular level. Pharmacology and pathology intersect intimately. Drugs treat disease, but disease alters how drugs act. For example, liver failure slows drug metabolism, prolonging effects and increasing toxicity risk. Kidney disease impairs excretion, allowing accumulation. Inflammation can change receptor sensitivity, modifying responses. Conversely, some drugs cause pathology, Certain chemotherapeutics damage bone marrow, some antibiotics injure kidneys, some anti-inflammatories erode stomach lining. Recognizing these intersections is crucial to safe application. Clinical examples illustrate: consider anticoagulants, drugs that prevent clot formation. They reduce risk of stroke in patients with irregular heart rhythm, yet excessive effect causes bleeding. Pathology explains the danger. Blood vessels cannot seal properly when injured. Monitoring and adjustment become essential, informed by both pharmacology and pathology. Or take tuberculosis. Pathology reveals granulomas, clusters of immune cells walling off bacteria. Pharmacology develops antibiotics that penetrate these lesions, targeting dormant organisms. Understanding one field enriches the other. Technological progress reshapes both. High-throughput screening identifies potential drugs among millions of compounds. Molecular pathology uses sequencing to identify mutations in tumors, guiding drug choice. Imaging mass spectrometry maps drug distribution within tissues. Artificial intelligence analyzes data to predict interactions and outcomes. These tools accelerate discovery, yet also highlight complexity. The body is not a static vessel but a dynamic ecosystem. Comparative studies widen scope. Plants produce alkaloids as defenses, many of which humans adapt as drugs. Digitalis from foxglove strengthens heart contraction. Morphine from poppy alleviates pain. Quinine from cinchona treats malaria. Each reflects pharmacology hidden in nature, uncovered through observation and refined through science. Likewise, pathology in animals informs human medicine. Studying prion diseases in sheep illuminated mechanisms of mad cow disease and Creutzfeldt-Jakob disease. Thus, knowledge crosses species boundaries. One metaphor captures the unity. Imagine a great library where some volumes record how the body works and others describe how it fails. Pharmacology reads the volumes of intervention, writing new chapters with synthetic compounds. Pathology reads the volumes of dysfunction, cataloging errors in script and structure. Together they tell the story of biology under pressure, whether from external molecules or internal breakdown. Reflection may come quietly every time you take a medicine or undergo a test. Centuries of accumulated pharmacological and pathological knowledge lie behind the act. The tablet in your hand and the slide under a pathologist's microscope are outcomes of disciplines that sought to understand both the power and the fragility of life. And yet, disease rarely confines itself to individuals. Outbreaks spread across families, cities, and nations, shaping history. This leads naturally to the next question: how do patterns of health and disease unfold, not in one body, but across entire populations? Health is never confined to a single individual. It spreads, shifts, and transforms across families, neighborhoods, nations, and even continents. When an illness arises, its course is shaped not only by biology within a body, but also by patterns of contact, environment, and behavior among populations. Epidemiology is the science that studies these patterns. It does not focus on a single patient, but instead asks how many are affected, where, when, and why. Through such questions, it seeks to understand disease at scale and to protect communities. At its foundation, epidemiology distinguishes between incidence and prevalence. Incidence counts new cases appearing in a defined period. Prevalence counts all cases present at a given moment. These measures reveal different aspects. A disease with high incidence but short duration may have low prevalence, like influenza. A disease with low incidence but long duration may have high prevalence, like diabetes. Both measures are essential for interpreting the burden of illness and planning responses. Patterns of disease occurrence are rarely random. Some diseases cluster in certain regions, others in specific age groups, still others during particular seasons. Cholera often flares in areas with poor sanitation. Malaria thrives where mosquitoes breed. Seasonal flu peaks in winter. By charting these patterns, epidemiologists generate hypotheses about transmission and risk factors. They then test these through analytic studies, comparing groups exposed and unexposed, diseased and healthy, to infer associations. One classic example is John Snow's investigation of cholera in 19th century London. By mapping cases, he identified a contaminated water pump as the source. His intervention, removing the pump handle, halted the outbreak, demonstrating that careful observation and mapping could save lives. Modern epidemiology uses far more sophisticated tools but retains the same essence, linking patterns in populations to causes and interventions. Transmission dynamics are central. Infectious diseases spread when susceptible individuals contact infectious ones, influenced by factors such as environment, behavior, and immunity. The basic reproduction number, R0, estimates how many new cases one case generates in a fully susceptible population. If R0 is greater than 1, an epidemic can grow. If less than 1, it dwindles. Vaccination, behavior changes, or treatment can lower effective transmission, bringing outbreaks under control. Non-infectious diseases also fall within epidemiology's scope. Cancer incidence varies with genetics, diet, and exposure to carcinogens. Cardiovascular disease relates to smoking, activity, and nutrition. Epidemiologists design cohort studies, following groups over time, or case control studies, comparing affected and unaffected to uncover associations. Such work established links between tobacco and lung cancer, high cholesterol and heart disease, ultraviolet light and skin cancer. In each case, population-level analysis revealed risks invisible at the individual scale. A metaphor helps. Imagine standing on a hill overlooking a forest. From close up, you see individual trees, each with unique shape and health. But from the hill, patterns emerge: dense clusters, fire scars, paths of disease through the canopy. Epidemiology takes this higher view. It steps back from the single patient to see how illness travels through communities, revealing causes that might remain hidden when looking only at individuals. Technology has transformed the field. Geographic information systems map outbreaks in real time. Sequencing tracks mutations of viruses as they spread globally. Statistical models simulate scenarios, predicting whether interventions will flatten curves or allow surges. During pandemics, data streams from hospitals, laboratories, and mobile devices merge, creating a living picture of transmission. Yet with data comes responsibility, ensuring privacy, accuracy, and equitable access to knowledge. Clinical contexts underscore its importance. When clusters of pneumonia appeared, epidemiologists investigated and recognized a new coronavirus. Their models predicted spread, guiding policy on distancing, masks, and vaccination. Though outcomes varied across nations, the framework of epidemiology provided essential tools to understand and mitigate the crisis. Without it, responses would have been blind, delayed, and less effective. Epidemiology also investigates outcomes of interventions. Randomized controlled trials, considered the gold standard, allocate participants to treatments or controls, then compare results. These trials determine whether a drug, vaccine, or policy truly works, distinguishing correlation from causation. Observational studies complement this, revealing long-term trends and rare effects not captured in trials. Together they form the evidence base for medicine and public health. But challenges persist. Diseases do not respect borders. Global travel spreads pathogens within hours. Climate change alters habitats of vectors like mosquitoes, expanding zones of malaria, dengue, and Zika. Urbanization, deforestation, and wildlife trade increase contact between humans and novel pathogens. Epidemiology adapts by strengthening surveillance, building networks of rapid response, and collaborating internationally. Still, uncertainty remains constant. Predicting the next outbreak is never exact. Chronic diseases pose different challenges. They develop slowly, influenced by multiple factors. Untangling diet, genetics, behavior, and environment requires massive datasets and careful statistical control. Yet the payoff is immense. Identifying risk factors allows prevention. Campaigns to reduce smoking, encourage exercise, or improve diet stem directly from epidemiological evidence, saving millions of lives even without specific cures. Comparative studies enrich understanding. Populations exposed to high altitude adapt physiologically, influencing oxygen delivery. Isolated groups with limited diets reveal effects of nutrient deficiencies. Migrant studies show how risk shifts when individuals move to new environments, separating genetic from cultural influences. These natural experiments, interpreted through epidemiology, uncover interactions between biology and lifestyle. Pathology may explain how a disease harms tissue, but epidemiology explains how it spreads across time and space. Pharmacology may show how a drug works in a body, but epidemiology reveals whether its use lowers mortality at scale. Thus, the field provides the essential bridge between individual biology and collective health. Reflection arises, each statistic in a report, one percentage point, one curve, represents countless human stories. Behind graphs are lives altered, families disrupted, futures changed. Epidemiology translates these stories into knowledge, knowledge into action, and action into protection. From here, another question beckons. If populations change over generations, how did the immense variety of living forms arise, and what deep history connects all organisms back to a common origin? Every species alive today carries within it a record of ancestry, written not only in genes, but also in bones, teeth, shells, and impressions left in stone. The study of these traces, paleontology, together with evolutionary biology, reveals the deep story of life on Earth. It is a story not of steady progress, but of branching, experimentation, extinction, and survival. Through it you come to see the continuity that binds bacteria, plants, animals, and humans to a single origin billions of years ago. Fossils are the primary witnesses. When organisms die, most vanish without trace, decomposed and recycled. But in rare circumstances, remains become buried in sediment, mineralized and preserved. Over immense time, layers of rock accumulate, and within them lie bones of dinosaurs, shells of mollusks, leaves of ferns, even delicate imprints of feathers or skin. Each fossil provides a snapshot, but many together form a motion picture spanning eons. Dating techniques, radiometric clocks, stratigraphic order, molecular calibrations place fossils in time, allowing reconstruction of evolutionary sequences. One of the most striking revelations from paleontology is that life has been punctuated by mass extinctions. 5 major ones are known: the Ordovician, Devonian, Permian, Triassic, and Cretaceous. Each eliminated large proportions of species, yet each was followed by diversification. The Permian extinction wiped out most marine life, but survivors radiated into new forms. The Cretaceous extinction ended the reign of non-avian dinosaurs, but opened niches that mammals later filled. These events illustrate evolution's rhythm, destruction followed by renewal, shaping the course of biodiversity. Evolutionary biology explains how such change occurs. At its foundation is variation. Individuals differ genetically. Some variations confer advantages in survival or reproduction. Natural selection favors these, while less useful traits fade. Over generations, populations shift, adapting to environments. Genetic drift, migration, and mutation add further forces. Speciation arises when populations become isolated, accumulating differences until they can no longer interbreed. The branching of species over time produces the tree of life, with every living form connected. Evidence for evolution is overwhelming and multifaceted. Fossil sequences show gradual transitions, reptile-like ancestors evolving mammalian features, fish developing limbs that became legs, feathered dinosaurs leading to birds. Comparative anatomy reveals homologous structures, human arms, whale fins, bat wings, all variations on the same skeletal theme. Molecular biology demonstrates genetic similarities. Humans share about 98% of their DNA with chimpanzees, and even with yeast share core genes essential for cellular processes. Such concordance across independent fields confirms a common ancestry. A metaphor illuminates: imagine a vast family tree stretching back through generations. Branches represent lineages, twigs represent species. Some branches end abruptly, species gone extinct. Others split repeatedly, producing clusters of diversity. The trunk represents the universal ancestor. Evolutionary biology is the discipline that reconstructs this tree, using fossils, genes, and morphology as clues, and showing that you, along with every other organism, are leaves on its outermost twigs. Consider human evolution as one branch. Fossils of Australopithecus reveal upright walking millions of years ago, long before large brains appeared. Later Homo species show increasing cranial capacity, tool use, and migration out of Africa. Genetic evidence supports this narrative. Modern humans trace ancestry to populations in Africa about 200,000 years ago, with limited interbreeding with Neanderthals and Denisovans. These discoveries remind us that humanity is recent, one branch among many, and shaped by the same evolutionary forces as all life. Pathology intersects here too. Genetic disorders sometimes trace back to ancient adaptations. The sickle cell mutation, harmful in double form, persists because carriers gain resistance to malaria. Lactose tolerance, rare historically, spread in populations practicing dairy farming, showing cultural practices feeding back into evolution. Even today, pathogens evolve resistance to drugs, exemplifying natural selection in real time. Thus, evolution is not confined to the past, but continues around and within you. Paleontology provides not only bones, but also insight into ecosystems. Fossilized pollen reveals climates, trackways reveal behavior, coprolites reveal diet. From these fragments, ancient worlds are reconstructed, seas of trilobites, forests of giant ferns, plains of mammoths. Evolution is contextual. Organisms adapt not in isolation, but within webs of interaction. Predators shape prey, plants shape pollinators, climate shapes distributions. By studying these dynamics, paleontologists and evolutionary biologists explain not only lineage but also ecology across time. Technological advances deepen resolution. CT scans peer into fossil interiors without destruction. Isotopic analysis reveals ancient diets. Molecular paleontology recovers fragments of DNA or proteins from extinct species, offering genetic insight. Computer simulations model evolutionary scenarios, testing how traits spread. These methods refine narratives once based only on bones into multidimensional reconstructions of ancient life. A clinical connection emerges in evolutionary medicine. Many traits that once enhanced survival now contribute to disease. Cravings for sugar and fat, once adaptive in scarce environments, now fuel obesity and diabetes. Back pain arises from spines adapted for quadrupedal ancestors, now bearing upright posture. Anxiety, once a useful vigilance against predators, can become maladaptive in modern settings. Recognizing these evolutionary mismatches clarifies why some illnesses persist despite medical progress. Reflection at this scale is profound. The stone in your hand may contain a fossil hundreds of millions of years old, connecting you to organisms long vanished. The genes in your cells carry echoes of survival strategies honed over billions of years. To study evolution and paleontology is to see yourself not as separate from nature, but as part of a continuum stretching back to the first replicating molecules. Yet another question arises naturally. If today's diversity arose from ancient branching, how do scientists classify the living forms around us, distinguishing plants, animals, fungi, and the countless creatures of sea and land? The living world today is astonishing in its variety. From towering redwoods to microscopic plankton, from vast herds of mammals to invisible fungi under soil, the spectrum of organisms reflects billions of years of diversification. To make sense of this abundance, biology divides into disciplines that focus on major groups: zoology for animals, botany for plants, and marine biology for life in the oceans. Each field classifies, observes, and interprets living forms, while recognizing that boundaries blur and interactions bind them together into larger systems. Zoology begins with animals, multicellular organisms that feed on others for energy, move at least during part of their life cycle, and sense their environment. Animals range from sponges lacking true tissues to mammals with complex nervous systems. Classification groups them into phyla, arthropoda, chordata, mollusca, and many more. Arthropods, including insects, spiders, and crustaceans, represent the most numerous and diverse group. Chordatids include vertebrates with backbones and internal skeletons ranging from fish to reptiles to birds and mammals. Each phylum reflects evolutionary innovation: hard exoskeletons, jointed limbs, jaws, feathers, or placentas. Within zoology, behavior is a central theme. Ethology studies how animals act in natural environments. Birds build nests with precise architecture. Ants cooperate to form colonies functioning like superorganisms. Dolphins communicate with whistles and clicks. Behavior emerges from both genetic programming and learning, shaped by survival needs. Comparative studies reveal convergences: bats and whales, though distant in lineage, both evolved echolocation, wolves and humans both developed cooperative hunting strategies. These parallels show how similar pressures lead to similar solutions. Physiology within zoology explores how animals function. Desert species conserve water through concentrated urine and nocturnal habits. Arctic species preserve heat with insulating blubber and countercurrent blood flow in limbs. Migratory birds adjust metabolism for long flights, while hibernating mammals slow heart rate and temperature to endure scarce winters. Each adaptation reflects fine-tuned responses to environment, offering insights relevant even to human biology. Botany turns to plants, organisms that harness sunlight through photosynthesis. Green pigments in chloroplasts capture light energy, converting carbon dioxide and water into sugars. Plants form the base of most food chains, producing oxygen and organic matter. Classification divides them into groups such as bryophytes, mosses, pteridophytes, ferns, gymnosperms, conifers, and angiosperms, flowering plants. Angiosperms dominate today, their flowers and fruits enabling efficient reproduction and dispersal. Structure in plants mirrors function. Roots anchor and absorb water and minerals. Stems provide support and transport. Leaves maximize surface for photosynthesis. Vascular tissues, xylem for water, phloem for sugars, form pipelines connecting roots to shoots. Reproductive structures diversify, cones and gymnosperms, flowers and angiosperms. Pollination strategies vary from wind dispersal to intricate relationships with insects, birds, or mammals. The orchid that mimics a female bee to attract males exemplifies evolutionary ingenuity. Plants also defend and communicate. Thorns deter herbivores, chemicals repel or poison, and volatile compounds released when leaves are chewed can warn nearby plants. Symbiosis with fungi, known as mycorrhizae, enhances nutrient uptake. Nitrogen-fixing bacteria in legumes enrich soil. These relationships show that plants are not passive but active participants in ecosystems, exchanging signals and resources. Fungi, often studied alongside plants, form another vast kingdom. They decompose organic matter, recycling nutrients. Some form mutualisms, like lichens that pair fungi with algae, colonizing harsh environments. Others cause disease, such as rusts in crops or infections in animals. Fungi also provide benefits. Yeast ferments bread and beverages, molds yield antibiotics, and mushrooms supply food. Their filaments, or hyphae, create networks that can span forests, sometimes called the wood wide web, transmitting nutrients and information among trees. Marine biology extends perspective to the oceans, covering more than 2/3 of Earth's surface. Marine environments host unique adaptations. Gills extract oxygen from water, swim bladders adjust buoyancy, bioluminescence produces light and darkness. Coral reefs teem with biodiversity, built by colonies of tiny animals in partnership with algae. Deep-sea vents support ecosystems without sunlight, relying instead on chemosynthetic bacteria. The ocean's vastness still conceals countless undiscovered species, reminding us that even now the catalogue of life remains incomplete. The metaphor of a library applies here. Each species is a book, filled with adaptations, behaviors, and histories. Zoology, botany, and marine biology are librarians, categorizing and reading these volumes, comparing themes, noting patterns. Yet the library is alive, with books constantly being written, revised, or lost. Extinction removes volumes forever, while evolution authors new ones. The task of biology is to preserve and interpret this dynamic library before too many volumes vanish unread. Clinical relevance surfaces even here. Many medicines derive from plants, fungi, or marine organisms. The rosy periwinkle gave compounds effective against childhood leukemia. A fungus yielded penicillin. Marine cone snails produce toxins, now studied as painkillers. Understanding biodiversity is thus not only ecological but medical. Hidden within species may lie solutions to human disease. Loss of habitats risks losing such potential forever. Comparative approaches deepen understanding. Studying hibernation in bears informs research on muscle preservation. Exploring regeneration in salamanders inspires hopes for human tissue repair. Observing photosynthetic efficiency in algae guides biofuel design. Each species is a model, offering lessons beyond its own survival. By cataloging and analyzing them, biology expands its toolbox for both knowledge and application. Reflection is inevitable. When you look at a tree, a bird, or a fish, you see not just an isolated organism but the outcome of millions of years of adaptation intertwined with countless others. The diversity you witness is fragile, dependent on balance and cooperation within environments. This recognition invites the next question. If species are parts of larger webs, how do entire communities of organisms interact with each other and with their environments, and what happens when those balances are disturbed? The living world is not a scattered collection of species, each existing in isolation, but rather a vast web of interactions. Organisms depend on one another for food, shelter, pollination, protection, and even regulation of populations. This network of relationships within specific environments is the domain of ecology, while environmental biology extends the view to include human influence, showing how delicate balances are disrupted or restored. To understand life fully, one must study not only individuals and species, but also the communities and ecosystems in which they exist. Ecology begins with levels of organization. At the foundation lie individual organisms, each with unique adaptations. Groups of the same species form populations, which interact through competition, cooperation, and reproduction. Multiple populations together form communities, where predator and prey, parasite and host, mutualist and competitor coexist. When communities interact with physical factors such as climate, soil, and water, they form ecosystems. Multiple ecosystems across regions compose biomes, such as deserts, grasslands, and rainforests. The entire biosphere encompasses all ecosystems on Earth. This hierarchy shows how local interactions scale into planetary processes. Energy flow is a central concept. Almost all energy entering ecosystems originates from the sun, captured by photosynthetic organisms, plants, algae, and some bacteria. These producers convert sunlight into chemical energy stored in sugars. Consumers, from herbivores to predators, transfer this energy up the food chain. Decomposers, fungi, and microbes break down waste and dead matter, recycling nutrients into soil and water. Without decomposers, ecosystems would collapse under accumulated debris. Energy flows one way, diminishing with each transfer, while nutrients cycle repeatedly. This dual pattern sustains ecosystems. Food webs illustrate complexity. A simple chain might show grass eaten by a rabbit, which is eaten by a fox. But in reality, rabbits may also eat shrubs, foxes may also prey on birds, and birds may scavenge from carcasses. Arrows cross and loop, forming webs of interdependence. Remove one species and ripples spread. The extinction of wolves in parts of North America allowed deer populations to explode, leading to overgrazing, soil erosion, and reduced plant diversity. Reintroducing wolves restored balance, demonstrating how predators regulate ecosystems. Population dynamics reveal further insights. Populations grow when births and immigration exceed deaths and emigration. Initially, growth may be exponential, but environmental limits, food, space, disease, predation, impose carrying capacity. Populations then fluctuate around equilibrium. Some species exhibit boom and bust cycles. Lynx and hare populations rise and fall together, driven by predator-prey interactions. Others stabilize through feedback mechanisms. Understanding these dynamics informs conservation and resource management. Ecology also studies symbiosis, intimate relationships between species. Mutualism benefits both partners, as in bees pollinating flowers or gut bacteria aiding digestion. Commensalism benefits one without harming the other, as when barnacles attach to whales. Parasitism benefits one at the expense of the other, as in tapeworms or mistletoe. Each form shapes evolution, driving adaptations and counter-adaptations. These relationships remind us that survival often depends not on independence but on connection. Environmental biology highlights human influence on these systems. Agriculture clears forests. Irrigation alters water cycles, and industry releases pollutants. Greenhouse gases trap heat, shifting climates, melting ice, and raising seas. Oceans absorb carbon dioxide, becoming more acidic, threatening coral reefs and shell-building organisms. Plastic waste accumulates in gyres, entering food chains, Species vanish at rates far exceeding natural background extinctions, leading some scientists to call the present era the sixth mass extinction. Yet humans also attempt repair. Conservation biology seeks to preserve endangered species, restore habitats, and maintain biodiversity. Protected areas, captive breeding, and ecological corridors help populations recover. Reforestation sequesters carbon, while renewable energy reduces emissions. Environmental biology measures these efforts, analyzing effectiveness and proposing improvements. The challenge is scale. Local actions matter, but global cooperation is essential to address climate and biodiversity crises. A metaphor clarifies: imagine an intricate clock with countless gears turning in synchrony. Remove one gear and others strain, remove several and the clock fails. Ecosystems are such clocks. Each species, no matter how small, contributes to function. Bees pollinate crops, earthworms aerate soil, phytoplankton produce oxygen. When these gears falter, consequences cascade. Ecology and environmental biology teach us to see the hidden gears and to recognize our dependence on their smooth turning. Clinical connections emerge subtly. Infectious diseases often link to environmental changes. Deforestation can bring humans into contact with new pathogens harbored by wildlife. Climate change expands ranges of mosquitoes, spreading malaria, dengue, and Zika into new regions. Pollution contributes to respiratory illness, cardiovascular disease, and cancer. Thus, ecology and environmental biology are not abstract, but directly tied to human health. Protecting environments protects populations. Technological advances aid study and protection. Satellite imagery tracks deforestation, glacial retreat, and ocean temperature. Remote sensors monitor animal movements. Genetic tools analyze environmental DNA, detecting species presence from water or soil samples. Computer models predict outcomes of policy choices, showing how emission reductions or land management alter future scenarios. These methods reveal both peril and possibility, guiding decisions. Comparative perspectives inspire action. Traditional ecological knowledge from indigenous cultures emphasizes respect, reciprocity, and long-term stewardship. Observations passed orally for generations align with scientific findings, highlighting sustainable relationships with land and sea. Integrating such wisdom with modern methods enriches strategies for conservation and sustainability. Reflection here becomes urgent. When you walk through a forest, hear birdsong, or breathe air from leaves, you participate in these webs. Your survival is inseparable from pollinators, decomposers, and climate stability. Recognizing this interdependence shifts perspective. The environment is not backdrop, but foundation. From this realization arises the final question: if life on Earth is so intricately interconnected, might similar webs exist beyond our planet, waiting to be discovered, and how do we search for them? The search for life beyond Earth is both scientific and philosophical. It asks whether the intricate systems of molecules, cells, organisms, and ecosystems you have followed could arise elsewhere in the universe. This field, known as astrobiology, does not assume answers, but investigates conditions under which life emerges, adapts, and persists. Its scope stretches from deep-sea vents to distant exoplanets, linking chemistry, biology, geology, and astronomy in a single quest. The first step is to define life on Earth. Life is characterized by organization, metabolism, growth, reproduction, response to stimuli, and evolution. Yet debates remain. Viruses replicate but only within hosts. Prions spread pathological shapes but lack genes. Artificial systems may mimic lifelike properties. Astrobiology therefore adopts a pragmatic approach: search for systems that maintain complexity, use energy, and evolve, even if they differ from terrestrial examples. Planetary conditions form the framework. Life requires energy sources, building blocks, and solvents. On Earth, sunlight, geothermal heat, and chemical gradients provide energy. Carbon forms versatile molecules, while water serves as universal solvent. Thus, when scientists search elsewhere, they prioritize planets or moons with liquid water, carbon chemistry, and energy gradients. Mars, Europa, and Enceladus are prime candidates within our solar system. Exoplanets and habitable zones of distant stars expand the horizon. Mars has long fascinated explorers. Evidence shows it once had rivers, lakes, and possibly oceans. Today it is cold and dry with thin atmosphere and radiation exposure, yet subsurface ice and occasional methane plumes hint at possible niches. Rovers analyze rocks for organic molecules, drill for buried samples, and measure atmospheric dynamics. Even if life never existed there, studying its geology teaches how planets evolve and lose habitability. Jupiter's moon Europa presents another opportunity. Beneath its icy crust lies a global ocean kept liquid by tidal heating. Hydrothermal vents at the seafloor may provide chemical energy, analogous to those that host rich ecosystems on Earth. Instruments search for plumes erupting through the ice, potentially carrying biosignatures. Similarly, Saturn's moon Enceladus ejects jets of water vapor containing organics, salts, and hydrogen, all ingredients of potential metabolism. These moons remind us that habitability does not require Earth-like surfaces, it may flourish under ice. Beyond the solar system, thousands of exoplanets orbit distant stars. Some lie in habitable zones where liquid water could exist. Transit spectroscopy measures starlight passing through their atmospheres, revealing gases such as oxygen, methane, or carbon dioxide. Combinations of these gases, especially in disequilibrium, may indicate biological processes. The challenge is immense. Signals are faint, distance is vast, but technology advances yearly, bringing closer the possibility of detecting distant biosignatures. Chemistry remains central. Life on Earth is carbon-based, but could alternatives exist? Silicon forms complex structures, though less versatile. Ammonia might serve as solvent instead of water in colder environments. Astrobiology considers such possibilities without assuming Earth's template is universal, yet carbon and water remain primary targets given their abundance and proven capacity for complexity. Extremophiles on Earth expand imagination. Microbes thrive in boiling hot springs, acidic rivers, salt-saturated lakes, radioactive waste, and deep rock miles beneath the surface. These organisms demonstrate that life is far more adaptable than once thought. Each discovery widens the potential habitats elsewhere. If bacteria survive in ice, Mars' polar caps are plausible refuges. If they metabolize sulfur near vents, Europa's ocean floor becomes conceivable. If they endure radiation, surface environments once dismissed may be re-examined. A metaphor helps. Think of the universe as a vast library, each planet or moon a sealed book. Most remain unread. Astrobiology is the discipline that opens these books, scanning for familiar alphabets of chemistry and unexpected scripts of alternative life. Some volumes may contain only blank pages, barren worlds, but others may hold chapters of biology written in forms unlike our own. Detection strategies balance direct and indirect approaches. Direct searches look for microbial fossils, organic molecules, or isotopic ratios characteristic of metabolism. Indirect searches seek atmospheric patterns, oxygen with methane for instance, that suggest continuous replenishment by life. Even radioastronomy participates, scanning for artificial signals that might indicate intelligent beings. While speculative, such efforts highlight the continuum from microbial beginnings to advanced civilizations. Clinical relevance may seem distant, yet the connection exists. Studying extremophiles informs biotechnology, yielding enzymes stable in heat or acidity, useful in medicine and industry. Planetary protection protocols designed to prevent contamination, mirror infection control. Sterile techniques ensure spacecraft neither harm alien ecosystems nor return with hazards. Thus, astrobiology shares principles with health sciences: caution, sterility, vigilance applied on planetary scales. Philosophical implications are profound. If life exists elsewhere, humanity is not unique but part of a cosmic pattern. If it does not, Earth becomes even more precious, a singular cradle in a vast desert. Both possibilities inspire humility. Either we share the universe with countless forms, or we bear the sole responsibility to sustain life in all its diversity. Reflection closes the arc. You began with molecules and primordial pools, traced through cells, tissues, organs, populations, ecosystems, and now stand at the threshold of the cosmos. Each level revealed complexity, cooperation, and fragility. Whether or not other worlds harbor life, the one you inhabit does in spectacular abundance. protecting it becomes not only a biological duty but a cosmic one. And so the question lingers beyond science. If life arose here, where else might it be writing its story at this very moment, under alien skies and oceans we have yet to see? You have journeyed across the full map of biology, from the smallest molecules to the vastness of cosmic possibility. At each step, complexity revealed itself, not as chaos but as patterns shaped by time, adaptation, and connection. Cells assembled into tissues, tissues into organs, organs into organisms, organisms into populations, populations into ecosystems, and ecosystems into the biosphere. Together they compose the single fragile phenomenon we call life. As you reflect, you may sense the quiet rhythm underlying all of this. Molecules interact without pause, cells divide and repair, organs sustain their tasks, and ecosystems exchange energy. Much of it happens in silence, beneath awareness, yet it continues tirelessly. The very fact that you breathe, think, and wonder is evidence of biology's continuous unfolding. There is humility in this realization. The sciences of anatomy, physiology, genetics, ecology, and beyond are not separate but intertwined perspectives on the same fabric. Each discipline illuminates one thread, but the tapestry becomes visible only when viewed together. Biology is not only the study of life, it is life studying itself, turning its gaze inward and outward, seeking to understand. And as the journey closes, you may hold one thought gently. Life is both resilient and delicate. It adapts across millennia, yet can be unsettled by a single change. To know it is to respect it, to recognize that every organism from microbe to whale shares the same long history and the same fragile future. In this awareness there is calm. You rest within the continuity of life, connected to its past and part of its unfolding story, carrying forward the curiosity that has guided this exploration.